Right now, inside every cell of your body, there are tiny molecular switches being flipped on and off. Some of them changed state this morning when you felt anxious about an email. Some shifted last night when you laughed until your stomach hurt. And some were set decades ago by experiences you no longer consciously remember. Your thoughts, your moods, your stress, your joy. They don't just live in your mind and vanish. They leave physical marks on your DNA, not on the sequence itself, but on something stranger and more powerful layered on top of it. Your genome is often described as a blueprint, but that metaphor undersells the situation. A better picture. Imagine a massive library containing every book ever written, packed floor to ceiling with volumes, and almost all of them chained shut. The books that get read in this specific cell on this specific day determine whether you become a neuron or a liver cell, whether you mount an immune response or stay quiet, whether you grow or hold still. Your DNA sequence, all 6 feet of it, coiled inside a nucleus the size of a dust speck, is that library. But sitting alongside every book is a second instruction manual, telling the librarian which volumes to open and which to ignore. This second layer of control is called epigenetics, from the Greek meaning above or on top of genetics. The epigenome doesn't change your genetic letters. It changes which letters get read, which sentences get spoken aloud into proteins, which chapters of your biological story get told at all. And unlike the genome, which is essentially fixed from conception, the epigenome responds to what you eat, to how much you sleep, to the quality of your relationships, to how your nervous system processes the events of your day. It is the biological interface between your inner life and your molecular machinery. When something frightens you, your brain doesn't just register an emotion and file it away. It launches a cascade. The amygdala fires. Cortisol and adrenaline surge into your bloodstream within seconds. Your heart rate climbs, your pupils dilate, and deep in the nucleus of your cells, something begins to change. Cortisol, small and fat-soluble, slides right through cell membranes like they're not there and drifts into the nucleus, where it binds to glucocorticoid receptors. These receptors then act as transcription factors, molecules that physically dock onto specific regions of DNA and either recruit the molecular machinery that reads genes or actively block it. One stress response can simultaneously silence dozens of genes involved in digestion, reproduction, and immune function, while amplifying genes that mobilize energy and sharpen focus. This makes perfect sense for a short-term threat. A predator, a car swerving toward you, a collapsing building. But the epigenome wasn't designed to distinguish between a lion and a toxic work environment. Chronic stress means chronic cortisol, and chronic cortisol means those epigenetic switches stay thrown in the emergency position, long after the emergency has passed. The chemistry of sustained anxiety looks remarkably similar at the molecular level to the chemistry of genuine danger. The epigenome has 2 main tools. The first is DNA methylation, where a small chemical tag called a methyl group, 1 carbon atom bonded to 3 hydrogens, gets attached directly to a DNA base, typically cytosine. When enough methyl groups cluster near the start of a gene, they act like a padlock on that book in the library. The machinery that reads DNA can't get traction. The gene goes silent. The second tool works on the proteins DNA wraps around, called histones. Your 6 feet of DNA doesn't float loose inside the nucleus. It coils around spool-shaped histone proteins, like thread around a bobbin, and then those spools cluster together into higher-order structures. Enzymes can add or remove chemical tags from these histones. Acetyl groups, methyl groups, phosphate groups. And depending on what gets added where, the DNA either loosens and opens for reading or tightens and closes off. Stress, trauma, meditation, exercise, starvation, joy, loneliness, and love all shift these patterns. Researchers at the Max Planck Institute found that even a single session of mindfulness meditation produces measurable changes in the methylation patterns of stress-related genes. The physical structure of your genome is subtly different after an hour of sitting quietly than it was before. For decades, biologists assumed that epigenetic marks were reset at conception, that your children start fresh, biologically speaking, regardless of what you experienced. That assumption turned out to be incomplete. The Dutch hunger winter of 1944 gave researchers an unwilling natural experiment. When Nazi forces cut off food supplies to the Netherlands, pregnant women carried their fetuses through months of severe starvation. Decades later, the children born during that famine showed elevated rates of obesity, diabetes, and cardiovascular disease. So did their children. The epigenetic adjustments made in response to starvation, cellular programs tuned to hoard energy, conserve fat, treat scarcity as the permanent condition of existence, was somehow transmitted across at least 2 generations. Animal studies confirmed the pattern with even more precision. When male mice are chronically stressed before mating, their offspring show stress dysregulation, and so do their offspring, despite never experiencing stress themselves. The mechanism appears to involve small RNA molecules and incompletely erased epigenetic marks that survive the reprogramming that normally occurs in sperm and eggs. Your body is in conversation not just with your own life experiences, but with the lives of people who came before you. Some of what you carry, biologically, is their unfinished business. If experiences can write damaging patterns into your epigenome, the obvious question is whether different experiences can write them back out again. The evidence suggests yes, with caveats. Exercise is one of the most reliably documented epigenetic interventions. A single bout of aerobic activity temporarily demethylates genes involved in muscle metabolism and neuroplasticity, and long-term exercise gradually reshapes methylation patterns across hundreds of sites in brain, muscle, and fat tissue. Antidepressants appear to work partly through epigenetic mechanisms, altering histone acetylation in regions of the brain involved in mood regulation. Early childhood nurturing permanently alters the methylation of stress response genes in ways that produce calmer, more resilient adults. A finding first established in rat pups, and since supported in human studies examining childhood adversity and adult health outcomes. Psychotherapy, particularly trauma-focused approaches, shows measurable epigenetic changes in war veterans with PTSD. The molecular machinery that transcribes your genes is listening to what you do, what you experience, who holds you, and who leaves you. The genome is not a fixed sentence. It reads more like a living document that keeps being edited by the life you lead. None of this means you can think your way out of cancer, or that positive affirmations rewrite your biology in some magical way, or that people who develop illness simply fail to feel the right things. The epigenome is one layer of an extraordinarily complex system, and the connections between specific thoughts, specific epigenetic marks, and specific health outcomes are real but probabilistic, cumulative, and entangled with genetics, environment, microbiome, random chance, and factors researchers haven't identified yet. What it does mean is that the boundary between mind and body, which once seemed like a clean conceptual line, is porous at the molecular level in ways that matter. Chronic loneliness is not just a feeling. Sustained compassion is not just a virtue. The quality of your inner life is also a biological environment that your cells are constantly adapting to. Over a lifetime, those adaptations accumulate. They shape gene expression. They influence which parts of your biological library get read, and in some cases, they pass forward into lives that haven't begun yet. The molecules don't care about the philosophical categories we've built between psychology and physiology. They just respond to what's actually happening inside you, faithfully, every day, one methyl group at a time.